Hello, and thank you for your attention while I discuss how understanding the population genomic structure of rare desert species is vital in low-impact solar energy planning. As I am speaking to you from Michigan State University, I would like to acknowledge that I am on the occupied ancestral, traditional, and contemporary lands of the Anishinaabe, Three Fires Confederacy of Ojibwe, Ottawa, and Potawatomi peoples. The university resides on land ceded in the 1819 Treaty of Saginaw. Now, to begin, arid lands, specifically in the desert southwest of the United States, have some of the highest solar energy potential in the world. As the demand for renewable energy grows, solar energy is appealing to developers as it is one of the cleanest, most efficient forms of alternative energy. As we can see by the graph in the upper right-hand corner of the screen, there has been an almost exponential increase in solar development across the United States. However, this is not without cost. Previous studies have shown various ecological changes due to the presence of solar facilities. However, the short and long-term consequences of these have remained largely unexplored to date. This is especially interesting simply because solar facilities are designed for eventual decommissioning and removal. This leads to questions such as, how does solar development affect desert communities? And, can desert communities, which are notoriously slow to recover from disturbance, adapt to these rapid changes? To approach these questions, we look to the Mojave Desert region of the United States, the hottest but smallest desert region in North America. Over 54% of the Mojave is contained within California, and 25% of that region's rare desert plants are endemic to the state, making the Mojave a biodiversity hotspot. In fact, it is thought that not all species, plants especially, have been characterized in the Mojave Desert. Narrowing even further, our study took place in the Ivanpah Valley here, in what was once considered pristine, core ecological habitat. However, before moving forward, I would like to acknowledge that the Ivanpah Valley is within the ancestral lands of the Southern Paiute and Western Shoshone peoples. I'd like to offer gratitude for the land itself, for those who have stewarded it for generations, and for the opportunity to study, learn, work, and be in community with this land. This brings us to the specific facility of this talk. The Ivanpah Solar Electricity Generating Station, or ISEGS, officially opened in 2014. This facility is truly unique, not only because it remains one of the largest facilities of its kind worldwide, but in that the developers chose to avoid grading the ground beneath the mirrors in much of the facility, leaving most of the plant life intact, but impacted. When site surveys occurred, this region was experiencing drought, and much of the native vegetation was dormant. However, after construction began, there was a wet year, and rare plants started emerging from within the area of the facility. One of these plants is Mojave milkweed, an Idiroparus perennial capable of clonal propagation and largely localized to drainages and washes. It is an important food source for many charismatic mammals, such as black-tailed jackrabbits, as well as insects, such as the western monarch and queen butterflies. Mojave milkweed is currently classified as common but declining across its range. However, in California, it is considered rare, with only 4 occurrences documented prior to identifying the populations featured in our study. Some efforts were made to relocate the plants, but ultimately failed. So, what the developers decided to do is that in every area where there were milkweed, they moved the heliostats, or solar mirrors, slightly to create halo-like gaps in the array, such as the one seen in the lower left-hand corner of the screen. This left the milkweed directly undisturbed. However, the plants are now growing in a sea of solar mirrors. It is unclear how this will affect the milkweed, which brings us to the main purpose of this study. First, since next to nothing is known about the population genetics of desert plants, we needed to determine the current population structure of the milkweed before we can determine how best to conserve it. Are there distinct populations? Is the facility at 3,500 acres large enough to overlay an entire population? Second, once we know what the structure of the milkweed population looks like, we can investigate what unique genetic diversity may be contained within the plants in ISEGS. This kind of information may prove valuable to solar facility designers, as the alternative design of ISEGS was not without financial burden. If we can show that the unique aspects of ISEGS are successful in mediating the impact of the facility, this may help influence the design of future facilities. To investigate this, we first went out to the Ivanpah Valley to collect samples. We went out at 2 separate time points to take tissue samples. If we saw a plant that was large enough to sample, we sampled it. We also recorded the coordinates of all plants we saw. This gave us 96 samples across 4 sites in 2015 and 137 samples across 2 sites in 2018. One of the sampling sites is in the lower right-hand corner of the screen, which is currently covered by my video capture. We had 3 natural or undisturbed sites as well as a large collection area within iCIGS itself. Following field collection, we extracted DNA and prepared the samples for Illumina sequencing using the BestRAD method, where a restriction enzyme is used to randomly chop the genome into smaller fragments so we can easily find single nucleotide polymorphisms. BestRAD separates the ragtag isolation and library preparation steps, which ultimately recovers more unique fragments. Once we demultiplex the reads, we align the resulting data to the common milkweed genome and use STAX for SNP discovery. Following extensive quality filtering, we retained 10,904 loci across all individuals from both sampling years. After this, I ran the sequence data through various analyses to determine population structure and genetic diversity across the sampling locations. Our population structure results are as follows. We performed 2 different clustering analyses under different assumptions. 4 clusters were best supported. However, at k equals 3, iCIGS, here on the far right-hand corner of the lower plot, as well as Bobcat, or iCIGS and Excelsior, combined at similar frequencies. The Umberti population consistently clustered first. Pairwise FST values for the sites also showed statistically significant genetic differentiation. To further tease out these genetic relationships, we performed a minimum spanning network analysis on the samples. This plot shows the results of the minimum spanning network, which compares the pairwise genetic differences between individuals and links them together based on those distances. What is really interesting about this figure is the fact that the iCIGS population appears central to the other population, which indicates that it served as an ancestral source of genes for the other populations. This is also supported by the location of some individuals from other sites nestled within the iCIGS cluster. However, it is important to note that the main difference in this analysis versus the DAPC previously shown is that this is based only on genetic distance, or how similar the individuals are to each other. DAPC allows for multiple dimensions to be considered, so more fine-scale factors such as individual allele contribution are considered. Moving on, we looked at several diversity metrics using a dataset with putative clones removed. The results of our private allele analysis were quite striking. There were roughly 2,500 private alleles in the 2015 samples, with many found in a single individual. For the combined dataset, there were about 1,500 private alleles, none of which were found in a single individual. As the additional sampling year included many individuals from both iCIGS and Umberti, an overall decrease in private alleles is not unexpected as the populations are geographically close to one another and would reasonably share many alleles. The fact that we see the opposite trend may indicate that we instead found a lot of cryptic diversity not found in the original sampling year due to many plants existing as rhizomes. This is further supported by the fact no private alleles were found in a single individual in the combined years, while there are many cases of a single individual containing an allele in the 2015 sampling. In summary, the milkweed within iCIGS is currently a distinct population with unique diversity. This also shows that the novel construction used in iCIGS was successful in preserving an important population of Mojave milkweed in the Ivanpah Valley, and will hopefully urge more developers to consider the population genetics of the plants within their construction sites for future projects. Finally, solar energy is growing, and it will be important to design facilities that ultimately mitigate the effects of this development. Lastly, I would like to acknowledge my fellow lab members as well as some really important contributors and sources of funding. And I would love to hear any input from you all, so feel free to contact me via my Twitter or through my website. And thank you all very, very much for your attention, and I hope you all take care.